Dear students, today we have to learn about the cell division. There are two kinds of cell division that occur in living things, that are mitosis and meiosis. Then mitosis occur in the body cells of organism. That is, when our body cell divide, the kinds of cell division that occur is mitosis. That is, in mitotic division, the same number of chromosome is maintained by the cell. When a cell divide into two, the daughter cell also will bear the same number of chromosome. That is happening in the mitotic type of cell division. That is, in mitosis, same chromosome number is maintained. We know that in human being we have 23 pairs of chromosome in our cells. That is, 46 is the number of chromosome in the human cells. Then after the cell division, each daughter cell will have the same chromosome number. That is, in all the cell we will have the 46 will be the chromosome number inside the body cells of organism. Then in meiosis, that kind of cell division that occur inside the reproductive organs of living things. In case of human beings, the reproductive cells are sperms and egg. The sperms are produced inside the testis of male and eggs are produced inside the ovaries of female. We know that the sperm and egg unite together form an individual. If they carry only half number of chromosome, then only its union can regain the same number of chromosome as the individual. Or if it is a human, the sperm must carry 23 and egg must carry 23. Then 23 plus 23, it can retain that same number as 46 chromosome number after the fusion of sperm and egg. So meiosis, mitotic division should maintain half number of chromosomes. So they are known as haploid cells. After the division by meiosis, the cell must be haploid. Then in this chapter we have to learn about the mitosis in detail. For your examination, you have to learn about the mitosis in detail. The mitosis, for the division of the cell, there are two processes. First, it's nucleus divide. That is known as karyokinesis. Division of the nucleus is known as karyokinesis. Then division of the cytoplasm is known as cytokinesis. Karyon means nucleus. So nuclear division is known as karyokinesis. Karyokinesis containing 4 stages, that are prophase, metaphase, anaphase and telophase are the 4 phases of cell division. The several stages are there for the prophase. Certain characteristics are there for the prophase. The chromosomes have become short and thick and are clearly visible inside the nucleus. We know that we cannot, if you observe a cell through the microscope, we cannot see the chromosome distinctively. At the time of cell division only, it will become, in normal stage, it will remain as chromatin fiber, extremely thin structures that we cannot see through the microscope. At the cell division, that chromatin fiber will become shrink and they will thick, then only thick and short, then only it is visible through the microscope. Then chromosome, in the stage of chromosome only, it can be visible through the microscope. So if you observe, we can see that the chromosome become short and thick and are clearly visible in the inside the nucleus. Then second point, you can notice that each chromosome has already duplicated to form two chromatid. That in the here you can see in the picture, you can see the duplicated chromosome. Really only half of it lengthwise, if we divide the two strands, we can see one was the real one, the other structure, one of the structure is duplicated structure. Duplicated, it make its own kind, like a mirror image, not image, really that will produce its own kind. That is the duplication mean. The chromosome add materials to its side and make its own kind. A duplication or replication of chromosome occur before the cell division. In the case of cell cycle also, you can see two smaller cells are formed first. After the cytokinesis, two small cells form. In the picture you can see the first growth phase, the smaller cell grow into larger in the growth phase 1. In the then the second phase, you can see the synthesis phase. More DNA is synthesized, chromosome duplicate. That second stage that happen. That before the prophase stage, more DNA is synthesized. Synthesis phase, you can see in the cell cycle, you can see chromosome duplicate but remain attached. Here centromere is the region. In the duplicated chromosome, you can see centromere, and two from the centromere you can see two strands are there in the upward position, and two strands are there in the downward. Longer two strands are there in the downward position. These each hand of that duplicated chromosome is known as chromatids. These each hand-like portion of this from that centromere, four hand-like portions are there. That duplicated each part is known as chromatid. Then again you can see the prophase. Certain stages are there for the prophase. The first, you know that you learned about the centrioles. The first the cell organelle that is starting the cell division, that are the centrioles. First they duplicate and starting to move apart to opposite pole. You can see then certain radiating microtubules are also starting, emerging or forming from it. You know that around the centriole there are microtubules. Already they have microtubules. Then these centrioles, they first duplicate, and that is the starting of the cell organelle. That start the cell division is that centrioles. Centrioles duplicated and they starting to move apart. Then certain asters, we can say star-like or radiating microbes, microtubules are also formed from the duplicated centrioles, and they are starting to move to the two opposite pole. That is the first stage of cell division, and it is one of the characteristics of prophase. Then again you can see that the chromosome become distinct. It will be visible if we look through a microscope. It will be visible, the chromosome will be visible, the duplicated, then the centromere, you can see the attached region is known as the centromere, and the chromatids, the duplicated chromosome, each hand is known as chromatid, that are clearly visible in the prophase stage. Then again, spindle fiber, another structure is known as it is the first stage of prophase. You can see early prophase, you can see two stages, early prophase and late prophase are the two stages. On one side it is shown the animal cell, and here on the right side a plant cell is shown there. You know that the plant cell will not contain the centrioles. The organelles known as the centrioles are not present in plant cell, so it cannot be seen in the plant cell. But the in animal cell we can see the centrioles. Then the centrioles produce another structure known as spindle fibers. You can see thread-like structures are coming from the centrioles, and it is connecting two opposite poles. You can see two opposite poles are connected by this spindle fibers, and sometimes you can see the spindle fibers attached at the region of centromere. You know that centromere is the portion of the chromosome where the chromatids are attached, that is the centromere. Then here the spindle fibers are attached at the centromere. Then another important feature of this prophase is that nuclear membrane. You can see the nucleus of the cell nucleus. You can see the nucleus of the cell will become or starting to disappear. You can see the nuclear membrane becoming disappear. That are the stages of prophase. Then sometimes there may be a question, write the characteristics of prophase. That is one of the questions that may be asked for the board examination. Prophase, first stage is duplicated centrioles starting to move to the opposite pole. Then again the chromosome, the duplicated chromosome we can see. Then another important feature is nuclear membrane starting to disappear. These are the important stages of prophase. I think you understand. If I taught everything or all the stages at a time, it may be difficult for you. So I am finishing today by prophase or teaching you the stages of prophase. I am finishing today's class. I shall give some question answer through WhatsApp. Thank you.